Okay, welcome back, all of you. Students, am I audible?
Yes, ma'am.
Okay. So now when we say yes, tell me. So when we say respiration, what is respiration? What is respiration, guys?
Process of breaking down.
Process of breakdown of the organic compounds. So where this organic compounds has come from? Where this organic compounds has come from? Online students, I want you people be active and give me the answer.
I'm from the ingested food.
From the food. So what is broken down? Which organic compound is broken down to release energy? Go on answering. These are all introduction which you already have learned in your previous years, especially in 10th standard. You would have learned a little more extensively this one. Online.
And glucose.
Glucose. Is it only glucose that is broken down during to give ATP, or even other substances also involved?
Glycogen can be broken down. Or in case of starvation, proteins can be broken down.
Okay. If starvation is not there,
Then either glucose or glycogen. No, wait, I think glycogen is in fasting.
yeah. It's not opening my annotated notes here. How come? Just a minute. It's not at all opening. Are you sure? Did you see that?
You need to download the app.
Oh my God. Okay. I will send you the annotated notes. So let's do the, yeah. Respiration. In plants. Okay. What is respiration? Quickly, Purvi.
Ma'am, the breakdown of organic compounds to release energy.
To release energy. Okay. And where is this energy trapped? So these are all introduction. Where this energy is trapped?
In ATP
It's
molecules.
trapped in. So energy is trapped in. Energy is trapped in. She's telling one more answer.
Trapped means it's stored.
Yeah. Energy is trapped in. Where? I'm just asking, all these you would have learned in your 10th standard.
ATP.
Where? Yeah, glucose, any organic substances in which, how is it trapped?
ATP molecules.
Loud.
ATP molecules.
No. Energy is trapped in these bonds, CC bonds, that's in the organic, that is carbon to carbon bonds in the organic compounds. And breaking of this organic compounds releases enormous energy, leading to the considered formation of energy. So that formation of energy, that energy is in the ATP molecule. So synthesis of ATP is the main objective of cellular respiration, right? Synthesis of ATP. It's the major funder of cellular respiration. Right? These respiratory substances, next. I'm just writing the gist of introduction. So which are the respiratory substances? Which are the respiratory substances?
Ma'am, like which help in respiration.
Respiratory substances are carbohydrates, proteins, and fats. So you know that these respiratory substances are fuels, nothing but fuels. Can we call it as fuel?
Yes.
Yes. So they will have fuel, any fuel will have a particular calorific value, right? So what is the calorific value of these fuel? What is the calorific value of these fuel? So it's about 4.1. So kilocalories per gram. Okay, and this is about 5.7 kilocalories per gram. And this is about 9.5 kilocalories per gram. So all of them, if you take calorific value of fat is double the calorific value of your carbohydrate. If you eat double, the same 1 spoon of fat will give you the higher calorie.
Yes.
Right? What is that calorific value means? The amount of heat, calorific value that you would have learned in your physics. So the amount of heat that is required. So what is calorific value? What is calorific value, Vihan? What is a calorie? Okay.
It's the amount of energy released.
The amount of heat required for the complete combustion, oxidation of the given substance to release. Okay, 1 gram of food in a closed chamber. What do you call that closed chamber? Where this is done? Warm calorimeter. Have you learned that? In physics, yes, you would have come across. So bomb calorimeter is filled with oxygen. So the complete combustion of oxidation of this organic compound in the bomb calorimeter is called as calorific value. Okay. Now, you know that why oxygen is used as this substance here. I will try to open here. It's not possible to post and do it. Hot water भी नहीं है मुझे hot water चाहिए पहले yeah.
Mom, is it because the oxygen is responsible for breathing?
Yes, so oxidation takes place, the complete oxidation takes place in that. So when we talk about this respiration, you know that there are two types, aerobic and anaerobic. So we are talking here about the cellular respiration. Guys, are you understanding online students?
Yes.
Online students? Yeah. So we are here in the cellular respiration. In this cellular respiration, so this is cytoplasm and these are mitochondria. Okay. So here glycolysis takes place. Glycolysis, that is where the glucose is broken down. So glycolysis is the first part. And here the further oxidation takes place. So the complete breakdown of the glucose to C6 compound to C3 compound, and then it gives rise to, it enters into the, this C3 compound enters into the mitochondria in the presence of oxygen. If there is no oxygen, so then this C3 compound releases C2H5OH. What is C2H5OH? Ethyl alcohol along with carbon dioxide, and 2 ATP is released. The ATP released is very less here. And here 36 ATP is released. Okay. And sometimes in your muscle, when there is inadequate supply of oxygen, so what is released? C3 is released. So it's converted to lactic acid. Right?
Both are C3.
Yeah. So now let's go to now. So glycolysis. So I will shift to the iPad and I will show you here because I'm not getting where I have kept this. Just a minute, students. Give me a minute. It was here.
You check in the data files also.
No, I just kept yesterday only. Suppose in case if it doesn't work, so okay.
Then it must be the reset file.
Quick search. Yes, I have to copy this.
You forward it with that.
Yeah, that's what. How will I forward?
Mom, you have Google Drive.
Yeah,
Mom, from that.
You can put it in the Google Drive and then you can.
Mom, or if you can connect both of them with WhatsApp.
WhatsApp.
WhatsApp. Mom, Google Drive.
What is that sound? Oh, you are doing that. This is the one which complete. So that we can go faster. If I write everything, it will be a slower process. I think I have got one. Let me see. Yeah. Yeah. So quickly go on answering the introductory part, all of you. Here. So let me ask you some questions on the introductory part. I want answer from first from Vihan, next Purvi, next Meda, next Advai, and then Swara. So let's go in this order. So please answer all the questions. So whatever I ask. When complete oxidation takes place. So it is aerobic or anaerobic. What do you call it as? Aerobic. So incomplete oxidation results in? Yeah, incomplete oxidation.
Someone else.
Lactic acid.
Lactic acid, anaerobic or ethyl alcohol. So respiration is an exergonic process. Why? Respiration is an exergonic process. Why?
Who should answer?
Answer, Vihan. When energy is released, it's called as exergonic process. When energy is consumed, it's called as endergonic process. Photosynthesis is an endergonic process and respiration is an exergonic process. So why? Because the breakage of that CC bond, the bonds between the organic compounds is broken down to release enormous amount of energy.
Mom, you are wearing through your laptop.
Yeah. So I'm just asking you the high general information about respiration before going to that. So next is what are the differences? Can one of you give me the differences between the aerobic and anaerobic respiration if it is given for 3 marks in your school exam? So what all you write?
Complete takes place in air.
The very first thing.
Oxygen is not in that, but it's.
Yeah. So further breakdown of that is glycolysis. It happens in both aerobic and anaerobic. Once the 6 carbon compound is reduced to 3 carbon compound, the further breakdown of the 3 carbon compound takes place in the cytoplasm itself in anaerobic, and in aerobic it happens in the mitochondria in the presence of oxygen. Okay, hence there is a complete combustion or complete oxidation. So whereas in anaerobic it is incomplete oxidation, right? So that's the next.
Mom, like anaerobic happens in lower organ.
Anaerobic happens in
Lower.
organism and yeah.
Sharing screen.
No, I'm not sharing screen. I'm just giving you these inputs which you have learned in your previous year, right? So can anyone give me the reaction of aerobic respiration? Chemical reaction. Equation.
C6H12O6 plus O2 gives CO2 plus H2 plus.
Yeah.
That's the.
So anaerobic respiration, anyone? Anaerobic respiration.
Mom, it produces ethanol.
Ethanol, yes, lactic acid in muscle, but ethanol, because it is an intramolecular respiration. Whereas this is in between the molecules happen. That is within the molecules, because it happens only in cytoplasm.
Mom, lactic acid itself is ethanol.
Yeah, oxidation of what actually happens? Oxidation of NADH happens. So here, oxidation of NADH, that means hydrogen is removed. Who accepts that hydrogen?
Water.
Come on, very good. Who accepts that hydrogen? Oxygen accepts that hydrogen to produce
Water.
water. Now give me the exact equation. Now give me the equation.
Mom, NADH plus O2 gives NAD plus H.
Where? That's not required now for equation. So what is the equation here?
Some glycolysis cycle.
Yes, it's here. So many things are opened. So is it possible for you to see this one PDF? Online? Students, I'm asking you.
Yes, ma'am.
So let me write the equation. What is the equation? Can you tell me? Vihan. Vihan, I want equation. Yeah, in the presence of oxygen gives CO2 and H2O.
Okay.
So now tell me, how much CO2, how much AT, this one, H2O?
Balance.
6 CO2,
Mom, this is with respect to it.
686 kilocalories. Okay. And 36 ATP.
Mom, in some scenarios, it's 40, 48.
No. Okay, so actually speaking, so this is how 6 water it is, how this water is given out. CO2 is given. So how this water is given out. So that's what let us understand now. So this is first step here is glycolysis. So I want you people open the glycolysis that's given in your NCERT. What all you have to write in your NCERT for that, I'll tell you so that it becomes easy for you to understand. Open NCERT, what is the page number? 229, page number. Okay, so have you all opened? All three of you?
Yes, ma'am.
Okay. So this is discovered by Emden, Mayerhoff and Parnas EMP pathway. So this is also called as EMP. So EMP is because of Emden, Mayerhoff, Parnas. So these scientists. It occurs in cytoplasm. Another important point, it's a universal pathway. That means any organisms, whether it is irrespective of whether it is aerobic or anaerobic, this is a pathway, the first step that always occurs in all the biological systems. In all the eukaryotes, this happens. Okay. So in this process, what happens actually? So glucose undergoes partial oxidation to form two molecules of pyruvic acid. Partial oxidation means what?
It doesn't completely.
That means partial oxidation. C6H12O6 will get hydrogen is removed from that partially. So glucose, you know that in plants glucose is from sucrose, right? Because from starch converted to sucrose, and sucrose is transported to different parts of the plant, and sucrose has glucose and fructose both. Hopefully you know that. So there is a that is conversion of the glucose to, okay, takes place. Interconversion of the glucose. Okay, invertase enzyme is there, so which helps in the conversion. So glycolysis has two phases, that is investment phase and payoff phase. So investment means what? Investment means what? So it has two phases further, priming phase and cleavage phase. Cleavage means breaking of that bond. So investment means you invest in something, business. So what you do? You first give the money. So that means ATP you take. Energy is not given, energy is taken and then pay off. So you put 10 rupees, you get 50 rupees. That's payoff. So that means whatever the minimum energy that is taken in the investment phase, so that is glucose is taken in. So which are the different phases here? So all of you open your NCERT and go to your glycolysis. I'll teach you this as stepwise, and I've written that the same way. Okay, so this is the stepwise which is given here. Okay, so this, there are 10 steps actually. Step number 1 is the first glucose. So glucose is a 6-carbon compound. It is written here. What has happened? Guys, are you getting to see the screen properly?
Yes, ma'am.
So you can just press on the corner. Yes.
Still buffering? But this goes off, no?
No, ma'am, it's share.
Okay. Anyways, they are able to see, that's all I want. So glucose is a 6-carbon compound here. Okay, and this glucose is converted to glucose 6-phosphate. So that means in the 6th phase, 6th glucose carbon atom phosphate group is added. And which is the enzyme always for phosphorylation? Kinase. So since glucose is a hexose molecule, it's called hexokinase. And which is the cofactor? Which is the cofactor? Magnesium is the cofactor. And what do you call that reaction? I have written all the details there. Look there. Phosphorylation. So this is called as allosteric inhibition. Why allosteric inhibition? What is the meaning of it? Go back to, I want you people tell me this, allosteric inhibition. I have taught you this in biomolecules. So now glucose 6-phosphate is converted to, I want you people tell me what is allosteric later. So glucose 6-phosphate is converted to fructose 6-phosphate. So glucose is also 6-hexose and fructose is also a hectose. Glucose is an aldose, fructose is a ketose, that's all. So that means both of them are the sugar molecules hexoses. So therefore conversion of one hexose to another hexose is called as isomerization.
Yes.
Okay, and enzyme is phosphohexoisomerase and it's a reversible reaction. Okay, so now fructose 6-phosphate is converted to, so here also it is 6C. Keep looking at 6C, which is given in, let me write it again, 6C here also. So now fructose 6-phosphate takes up one more glucose, phosphate, ATP, and is converted to the first carbon atom and the 6th carbon atom. So fructose 1,6-bisphosphate is formed. And hence this is also a phosphorylation. So phosphorylation, isomerization, phosphorylation. So which is the enzyme again here? Kinase. If you forget anything, kinase, if you write also you'll get marks. Remember for phosphorylation. So now fructose 1,6-phosphorylation, which is a 6-carbon atom compound, is converted to 2 3-carbon compounds, converted to 2 molecules of DHAP. What is DHAP? Dihydroxyacetone 3-phosphate. Okay, dihydroxyacetone phosphate, which is a 3-carbon compound, and since fructose 1,6-bisphosphate is split into this 2 molecules, so it's called as cleavage. Is it clear? So DHAP and G3P. G3P, where do you come across? G3P pool. Have you heard G3P anytime? Glyceraldehyde 3-phosphate, you have not heard?
Yes.
Where? 3-PGA, phosphoglyceraldehyde. Phosphoglyceraldehyde, the same thing.
Yes, Mama.
Where? Online students, where? Photosynthesis,
Photosynthesis.
where in photosynthesis? Kreb cycle. Isn't it? So that is Kreb cycle you have heard. Excellent. Kreb cycle I have not taught, my dear.
Carboxylation.
Where have you heard? Oh my God.
Carboxylation.
And Calvin cycle.
Calvin cycle.
Oh,
How Krebs cycle?
Yes.
Already you are saying Krebs cycle. So that means it is learned and not required now. Krebs cycle comes in respiration. So this is that's why I didn't teach you in between. Soon after that. So this is DHAP is converted to G3P. So this is a reversal of glycolysis I had told you there. Okay. Carboxylation, reduction, regeneration. Do you remember?
yes.
Yes. So here it's an isomerase. That means this can be converted to that, that can be converted to this. So what is that fun? Basically what we have understood. So fructose 1,6-phosphate is cleaved to form 2 molecules of triose. Which is the triose? Glyceraldehyde 3-phosphate. It's an aldose, aldehyde, aldose. So this glyceraldehyde 3-phosphate, okay, so is a 3-carbon atom, and this glyceraldehyde 3-phosphate, 3-carbon compound, sorry, and it undergoes oxidation. What is oxidized here? It gives hydrogen. So when hydrogen is added to acid, it becomes aldehyde. When from the aldehyde, hydrogen is removed, what it becomes? It becomes acid. Acid becomes aldehyde when you add hydrogen. When you remove hydrogen,
Should it become keto?
from aldehyde, when you remove hydrogen, it becomes acid. Glyceraldehyde phosphate dehydrogenase. So what is removed there? Hydrogen is removed. So where that hydrogen is added to? It is added to NADH. NADP to form NADP, NADH and H plus ion. Okay. And here inorganic phosphate is added. Inorganic phosphate is on it. So now glyceraldehyde 3-phosphate becomes 1,3-diphosphoglyceric acid or glycerate, one and the same. So here NAD becomes NADH. So what is NAD? Nicotinamide adenine dinucleotide is an enzyme derived coenzyme. So when coenzyme is reduced, it acts as an ATP precursor. So one molecule of NADH gives rise to 2 molecules of ATP. It's just like a currency. Now you are in India, so you can spend the money in rupees only. But if somebody gives you, your uncle comes from, let's say, London. So her aunt comes from Dubai. So somebody, Purvi's brother comes from, let's say, Japan. So she gets yen, you get pounds, you get dollars, someone gets dirhams. So will you throw it away because it is not used in India? No. You will keep it and you will get it converted. So what are they? They are the money precursor, right? Currency precursor. Similarly, this is also energy precursor. So they are all coenzymes. We have learned that in biomolecules. So these energy precursors, so when they are reduced, they are kept it there. So the first energy precursor formed here is due to that is oxidation, removal of the hydrogen from glyceraldehyde to form glyceric acid. So here there is that is IP, that is inorganic phosphate is also added, and hence you get 1,3-diphosphoglyceric acid. So this glyceric acid is converted by the removal of, what is that? ATP is removed, phosphate is removed.
Yes.
Right? And it is added to ADP. ADP becomes ATP. So every time why I have written 2 here? Anybody? I have written 2 ATP, 2 NAD. Why?
And because there's
Why?
molecules of 1,3-diphosphoglycerate.
Because fructose 1,6-diphosphate glycerol that is give rise to 2 molecules of 3 carbon. Remember that. So now onwards, everything 2 here. Okay, after cleavage. So here 2 molecules of ATP is converted to. So here it is what is SLP I have written here. Substrate level phosphorylation. What is substrate level phosphorylation? This is the first substrate level phosphorylation. A particular substance. Okay. So substrate level means from the particular substance, phosphate is removed and added and you are getting phosphorylation. That's called as substrate level phosphorylation, where oxygen is not used here directly. So this is because of the kinase. Which kinase? Phosphoglycerate kinase. So everywhere it is 3 carbon compound. So now 3-phosphoglyceric acid is converted to 2-phosphoglyceric acid. Okay, so that's due to isomerization. 2,3-phospho becomes 2-phospho. That's isomerization. Phosphoglyceromutase. Okay, 2-phosphoglyceric acid will become 2-phosphoenol pyruvate. So that is enolase, and there is a removal of hydrogen, and hence this particular step is dehydration. So fluoride can inhibit this reaction. So fluoride inhibitor reaction, this enolase. Okay, so this phosphoenol, enolase, because enol pyruvate it becomes, enol is added, becomes just pyruvate, where by the removal of 2-phospho becomes only pyruvate. Why? Both the phosphate is removed 2-phospho. That means at the 2nd position, phosphate is there. That phosphate is removed. Removal of phosphate again, a phosphorylation. So this is the second substrate level phosphorylation. So from the substance, phosphate is removed and pyruvate is formed. So in the end, so there are 2 pyruvate is formed. 3 carbon, that is. 2 pyruvate from 1 glucose. So come on, anybody, can you give me the complete step? Give me the complete step. Which is the first step?
Glucose.
Phosphorylation. of glucose.
Which is the first step?
Phosphorylation.
Phosphorylation, where there is a phosphorylation is both the ways it is phosphorylation. What phosphorylation is this?
On glucose.
Glucose to glucose.
ATP utilization. Glucose to glucose 1,6 diphosphate. So glucose to glucose 6 phosphate. Glucose to fructose 6 phosphate. Fructose to again phosphorylation. So 1,6. So how many the ATP is utilized in the beginning? 2 ATP. So how many ATPs are formed? Forget NAD. How many ATPs are formed in the end?
4.
How 4? 2 substrate level phosphorylation. Every time 2 ATP. So 4 are formed. So 4 minus 2 which is used. So now you have put 2 rupees and got 4 rupees. What is the net profit?
2.
2 rupees. Same thing here. Got it?
Yes.
All of you? So 4 ATPs utilized and 2 ATPs are used and net gain of ATP is 2 ATP. Similarly, how many NAD is produced?
2.
2. So 1 ATP, I told you 1 NADH produce 2 ATP. So 2 NADH means 4 ATP. So what is the net gain in glycolysis? ATP 6 ATP. So if you have the net equation of this glycolysis is C6H12O6 with the help of, so you require 2 ADP, 2 inorganic phosphate plus 2 NAD to convert it to CH3COCOOH. What is the CH3COCOOH? Pyruvic acid. COOH acid. CH3COCOOH. Okay. And 2 ATP is formed, 2 NADH is formed. So 2 plus 4, 6. And 2 hydrogen is released. 2 NADH and 2 H plus 2 H2O. So that hydrogen is formed, water. So in the end, so CH3COCOOH. Now, this CH3COCOOH pyruvic acid in other organisms which are anaerobic, so they form different substances. So there are, based on this, we have ethyl alcoholic fermentation, acetic acid fermentation, right? Lactic acid fermentation in different organisms. So let's understand this fermentation funda, how it happens. So you need to remember this, that's all. Which are the steps involved? Okay, one of you online student, Purvi, come on, give me the steps here. All the 10 steps you give me. Purvi. 10 steps that are involved in glycolysis. There are 10 steps in your textbook. You can look into your textbook and give me the name of the steps. I have given the name to all the steps. First step, phosphorylation. Second step, third step, fourth step,
Hydrogen.
cleavage. Cleavage is the step. Fifth step. Which is the fifth step? No, where phosphorylation, isomerization. Again, aldol. So cleavage is the fourth step. In the fifth step, isomerization. DHAP is converted to PGA,
Yes.
phosphoglyceric acid, 3-PGA. So now 3-PGA are G3P, one and the same. Okay. So now 3-PGA is converted to sixth step, oxidation, that is removal of hydrogen. When hydrogen is removed, it becomes acid. So, and there is API, inorganic phosphate is also added. So therefore, that is a oxidation, then phosphorylation, then substrate level phosphorylation, that is isomerization again. So after isomerization, dehydration, that is hydrogen is removed. When hydrogen is removed, so then what happens? Enolation. Enolation means it becomes eno. So that enolation is second substrate level phosphorylation. Last step is, what is that?
Phosphorylation.
Phosphorylation. So here 2 phosphorylation, there 2 phosphorylation, but here 4 ATP is given out. And dehydrogenation, oxidation, removal of hydrogen is oxidation. In that oxidation to NADP. So hence 6 ATP together. So now this, that is Gay-Lussac, is a person who discovered this fermentation. And this fermentation is demonstrated by Cuhne in the Cuhne's flask. So Louis Pasteur is the first. Louis Pasteur is the first person to observe this fermentation and called it as an anaerobic respiration. So this donates the harvest of the electrons to other inorganic substances. So now, which are those organisms that undergo this aerobic respiration? This is your pre-biological classification. We have learned it and it is cited here. Methanogens. What are methanogens? What are methanogens? Which group you add them to?
Archaebacteria.
Very good. Archaebacteria. They use this carbon dioxide as electron acceptor and reduce that carbon dioxide to CO2. Okay, you reduce carbon dioxide to? You do not reduce. You oxidize. Remember. So that's why O2 is given out. Okay, there. Sorry, O2, water is given out. O2 is reduced to form water. So here carbon dioxide is reduced to methane. Okay. So you oxygen, in the presence of oxygen, what is given out? Water and carbon dioxide. Carbon dioxide which is produced from?
Carbohydrates.
Organic, yeah, carbohydrates, that's right. So sulfur bacteria, what happens in sulfur bacteria? Sulfur dioxide is reduced to hydrogen sulfide. So that's why wherever sulfur bacteria is there, you get that stinking smell. So rotten egg smell, hopefully you know that H2S is a rotten egg smell. Okay, so next is what is the fate of this pyruvate that's produced by? This is the complete picture I have given here. This is the beautiful image which I could get it from some publisher book. So please look into this. The fate of pyruvate is given in this picture everywhere. What's the fate of pyruvate? Once pyruvate is formed in the cytoplasm. So in anaerobic respiration, the further breakdown of this pyruvate takes place there only. So I want you people take a keen look about it. You can see here, which color is better?
Red.
Yeah.
So here, CH3, CO, COOH, CH3, CO, COO, right? So this is a pyruvate. So it is pyruvate is C3H4O3, right? C3H4O3, CH3CO, So this pyruvate, okay? This is pyruvate. What is happening? Anaerobic respiration lactic acid. So lactate dehydrogenase. In the presence of lactate dehydrogenase, it takes the NADH and it is oxidized here. NADH is not reduced. So whatever the NADH that's formed, when it is formed? During glycolysis is utilized. So dehydrogenase means
Removal of
from NAD. So utilizing that, they form lactate. C3H6O3. CH3COOH becomes CH. How many H is added here? Look there. 2H is added here.
yes.
Okay. 2H comes from NADH. So you know that 2H is formed there. Whatever the H, NADH, which is an energy precursor, it is formed. It is utilized here to form lactic acid. Because energy is utilized, So it produces very meager energy. Meager. Same thing happens in glycolytic fermentation here. You see here, all of you. So decarboxylation takes place first. What is decarboxylation? Removal of carbon dioxide. So when removal of carbon dioxide, CO2, when it is removed, so then it forms CH3COH. What is COH group? Aldehyde group. So this is acetaldehyde. CH3COH is acetaldehyde.
No, it is CHO group, COH as alpha.
CH3CHO, right? What is that? CH3COH, CH group, acetaldehyde. CH3, C2H5, CHO is
CH3,
CH3, CHO is acetaldehyde. What is C2H5CHO?
formaldehyde.
Formaldehyde. That's right. Okay. So this is acetaldehyde. And here also, whatever energy utilized, is used here. So here there is a dehydrogenase here. So acetaldehyde dehydrogenase. And what is formed finally? What is formed? Ethanol, C2H5OH. So which is nothing but C2H4O2. So H is added. Here also C3H4O3. Here C3H4O3. So H is added here. So this is called as alcoholic fermentation. So why? C2H5OH is formed. Okay, alcohol is formed. So all these yeast, wine, everything is made like this. Okay, but what happens whenever in other organisms, whenever there is oxygen or whenever the pyruvate enters into the mitochondria. So that is aerobic oxidation. The same pyruvate, okay, utilizes that NAD what is formed, NAD. Okay, so what it forms here? So in all the other case, dehydrogenase is happening. Acetaldehyde dehydrogenase is happening here. Can you see?
Yes.
Here also dehydrogenase. What is forming here?
Ma'am, in this NAD and
Oxidation is formed. From NAD, H is removed and is added. So from this pyruvate, H is removed. See, what is C3H4O3, correct? So C3H4O3. What is happening? CH3H4, CH3 only remaining is here. So whatever the H that is removed is utilized here and CO2 is also given out.
No, CO2 is that upper branch.
Yeah, so what is added here? Coenzyme A is added. So acetyl CoA, CH3, C instead of CHO, it is CO. Okay, acetyl CoA, sulfur acetyl CoA is added. So this acetyl CoA enters into the Krebs cycle. Okay, so here also I have shown this here like this. So pyruvic acid forms acetaldehyde, decarboxylation, and now reduction. So this NADH becomes NAD, but that hydrogen is utilized to form ethanol. So more than 13% of the alcohol it is formed in yeast. Yeast dies. Too much of alcohol kills. Alcohol is act as antibacterial agent. Hopefully you know.
yes.
Yes.
Whatever they use, that sanitizer, Savlon, whatever, alcohol is there. In sanitizer more, 90% and above.
Yes.
So that's why sanitizers are used. So cleaning of anything happens through alcohol in hospitals.
Yes.
Hopefully you know, spirit is an alcohol. So 13% of alcohol proves to be fatal for yeast. Remember this. Okay. So that is alcoholic fermentation. So now oxidation of. So how this pyruvate oxidize further? See, this particular cycle begins, you can see in this box, whatever is written there. Cycle begins when a 2-carbon unit from acetyl-CoA reacts with a 4-carbon molecule oxaloacetic acid to produce citrate. So citrate is the first compound that is formed here. So 2-pyruvate, which is formed through, like once again I'm telling, 2-pyruvic acid, which is formed, I'm showing here like this. So see here, this is 2-pyruvate, which is formed during glycolysis, isn't it? C3H4. So removal of decarboxylation takes place. Carbon dioxide is removed and it is added to acetyl-CoA, sulfur acyl acetyl-CoA to form what? So this acetyl-CoA. So carbon dioxide is removed. CO2 when it is removed, what is left? What is left? See here, I have shown in this very clearly. Carbon dioxide is removed here. Can you see the carbon dioxide? So when that is removed, what is left? C3H4O3. CO2 is removed. So now hydrogen is also removed. So then carboxylation, dehydrogenation. So when hydrogen is removed from NAD,
Yes.
so it forms NADH, which is an energy precursor. So for every 1 pyruvate, 2 is removed.
Yes.
Right? For each of them, 1 is removed. So what is remaining there? C3H4O3, hydrogen is removed, CO2 is removed. So what is left now? CO is left. Remember, I mean, understand here. Where shall I write here? C3H4O3. CO2 is removed. Then what is removed? 1C is removed. O2 is removed. C double bond O. Then 1 C is there here. CH. CH3 is acetyl. Right. So this is coenzyme. So acetyl-CoA. Now it is called as acetyl. Coenzyme is added. So now it's called as acetyl. CH3CO is acetyl. Acetyl coenzyme A. It's called as. Got it now? So because of decarboxylation and removal of hydrogen, acetyl-CoA is formed. So this acetyl-CoA enters into the mitochondria. Suppose if there are other proteins and fats also. So how do they enter? So they also enter as acetyl-CoA and this is called as beta oxidation. Breakdown of the fat to acetyl-CoA is called beta oxidation process. Okay, so this is beta oxidation and here pyruvate dehydrogenase, 60 subunits. See, look here. Protein molecule enters into the Kreb cycle by the conversion of that protein to acetyl-CoA. So that means whether it is glucose, acetyl-CoA. So that means any organic matter. So acetyl-CoA is a sugar.
Yes.
Excess proteins and excess fats converted to sugar, and that's called as gluconeogenesis. Where it happens in your liver. Suppose if a person is taking too much of proteins, ketone body increases in the body. Ketone is nothing but ketose. Then it becomes ketose. CO group. Why ketose body? This is what. All of them are converted to glucose. Too much of fat.
Yes.
All of them are converted to glucose. Finally, that's why a person, obese person having too much of fat and all these diabetes increase. Why? Because this one. Okay, so they will, so everything is converted to glucose finally. So now it enters into the Krebs cycle. So now I want you people look here, not into your book at all. So in your book, the Krebs cycle is very, very meager.
Yes.
Look into the Krebs cycle in your textbook first, all of you. How many steps are they given? That one.
On this one.
Ah,
On the citric acid.
citric acid is only Krebs cycle. I will tell you that one.
Yes. Moments. Got that.
How many steps? Count.
One.
There are exactly 10 steps involved here also. So Krebs cycle is called as citric acid cycle. Why? The first stable compound found is citrate. Citrate is citric acid. Pyruvate is pyruvic acid. Hopefully you know that. Okay. So citric acid cycle. And why is it called as tricarboxylic acid?
Because three carbon.
3 carbon. Okay. So this is tricarboxylic. Carboxylic means what? COOH. Okay. So, and it's also called as Kreb. Why? Yes. Hans, Sir Hans Kreb. Okay. So all of you now look into your citric acid cycle and write certain points. I have written the steps involved in citric acid cycle, one below another. Before going into the citric acid cycle, I would suggest you to look here. So here, Sir Hans Kreb described this. So pyruvate dehydrogenase is a complex substance. Why is it required? Removal of the pyruvate dehydrogenase and pyruvate decarboxylase. So from the pyruvate, hydrogen is removed, carbon dioxide is removed,
Yes.
right? And then it becomes acetyl-CoA. So this is a concept all about this particular enzyme. This is a complex enzyme which is having 60 subunits. Subunits are 60 in it. So coenzyme TPP, dihydroxylipoyl transacetylase, okay, dihydrolipoyl dehydrogenase, and prosthetic group are FAD, TPP, NAD, FAD, all these are different. So you need not remember all these. You need to remember it's the very complex enzymes that's there in your body. So which it conversion of pyruvate to coenzyme A. So during that, NAD is converted to, see here. So this particular, this one you remember. Is it really?
Yes.
This you remember. So pyruvate plus 2 NAD plus 2 COA, acetyl-CoA. With the removal of CO2 and pyruvate dehydrogenase, you get 2 acetyl-CoA plus 2 NADH plus hydrogen ion and CO2.
Yes.
Is that clear? Now, so I'm sending this, do not worry. So the first step in your textbook, look into that. What is the first step? Who accept this 2 acetyl-CoA? I will go on asking the answer. So first person to answer is Purvi, because Purvi doesn't say no, I don't want to answer, or doesn't mute her microphone. I'm happy about it, frankly speaking. So that's why I would begin with her, from her. Yes, Purvi. Beta,
Yes,
can you
ma'am.
answer? Yes.
Yes, ma'am.
Acetyl. So every time 2 I am writing. Why? Because 2 pyruvate, 2 acetyl-CoA. So the cycle it becomes easy for you to understand the cycle. Okay. Acetyl-CoA combines with Oxaloacetic acid. Now, acetyl-CoA, how many carbon is there?
4.
2 carbon. It's a 2 carbon compound. It combines with oxaloacetic acid. Oxaloacetic acid had come in the C4 cycle also. Hopefully you remember the first carbon.
OA.
Yeah, OA. So how many carbon compound it is?
4.
Carbon.
4C. So 4C and 2C. What do you get?
6 carbon.
6 carbon compound. 6 carbon compound is which one? Citric acid. So hence this cycle is called as
Citric acid cycle.
citric acid cycle. And now once the citric acid is formed, coenzyme A is a coenzyme that is removed, that is released. And here water is also added for the formation of, so hence this particular reaction is citric acid synthesis. Synthase is the enzyme. Next is step 2 is dehydration. Dehydration means
Taking hydrogen.
removal of that hydrogen from citric acid. So hydrogen is water is taken, hydration is water. So you get cis aconitate. Cis aconitic acid. Is it there? I don't think that particular step is given. So you can add that step. So that's called as aconitase is the enzyme. Cis aconitase. Is it given? Please take the sharp pencil and all of you write. That is dehydration step. Next step. I want Vihan answer. Vihan, are you there?
Also, aconitate is the produce
Product from
product.
citric acid. After the removal of
Add water.
water. Hydration means water. Dehydration means water. Hydration means adding. Dehydrate means removing. So
Hydrogen.
why this step is not given? Because dehydration and hydration happens here. That's why it is not given in your textbook. So this is in like a forming one form to another form. That's all. So that's why this step is not considered important and is not given in your textbook. So now cis aconitic acid. Again, water is removed and it forms isocitrate. It's only the isomerization. Okay, isomer form. So this isocitric acid, now the very important concept, oxidation. So put important or highlight that step which is given in your textbook, 100% it will be given, oxidation. So here isocitric acid, Vihan answer, it will be given.
Isocitrate forms oxalosuccinate.
Yeah, it should be given, no? Please write this. What is given in your textbook? Show me once again.
Directly after citric acid, it goes to aspartate.
After citric acid, it goes to
Then succinic acid is given, but that is the next conversion of.
okay. So then isocitric acid. Okay, so with that is oxidation, isocitric acid becomes oxalosuccinic acid, where NAD forms NADH and H. So this is the first energy precursor is formed. How many are formed? Keep counting. Is it given in your textbook? Oxalosuccinic acid. But this is an important, very important step. Please add on. So citric acid, isocitric acid forms dehydrogenase of the citric acid to form succinic acid. What succinic? Oxaloacetic acid, now oxalosuccinic acid. There is a formation of NADH. Okay, here all the three acids, citric acid, aconitic acid, oxalosuccinic acid are COOH group. Okay, the organic acids, they have carboxylic group, and hence this cycle is also called as the first three which is formed. Which are they? Citric acid, cis aconitic acid, and succinic acid. So all the three are COOH group, and hence this cycle is called as tricarboxylic acid cycle. Clear? So now the dehydrogenation. So now oxalosuccinic acid, removal of the carbon dioxide takes place. See here, how many times the carbon dioxide is removed? That's another question. So we give out carbon dioxide during respiration. That's the reason. So that is oxalosuccinic acid decarboxylation to form alpha ketogluteric acid. Removal of succinic acid becomes glutaric acid. Alpha ketogluteric acid. So here the enzyme is oxalosuccinic carboxylase. Okay. So here again oxidation. You can see the oxidation. So oxidation first happened there. That's the first step. Important step. How come book is missed out giving it? Third oxidation I wrote. This is the fourth oxidation. Where all oxidation has happened? First oxidation, where it happened? Where the first oxidation happened? Anybody, have you noticed? Let me see. In glycolysis, second oxidation, pyruvic acid to coenzyme A.
Yes.
Third,
Yes.
please keep counting this oxidation, guys.
Yes.
Where? That's correct. So where is second? I'm asking you.
Second is.
Where is second? Did you notice here?
Mamco. Yeah.
Coenzyme A formation. Did you notice here? This is the second. First in glycolysis, second is here to NADP, NADH, and third is during the isocitric acid to oxalosuccinic acid. So please keep writing this. So I have written that as a heading. And fourth is here where alpha ketogluteric acid is converted to succinyl-CoA, and here coenzyme A is also added there. Okay, so this is the fourth oxidation, and here decarboxylation also takes place. Carbon dioxide is again given out. So now we will take a count of how many times carbon dioxide given out. Okay, so decarboxylation takes place and succinyl coenzyme becomes succinic acid. So succinothiiokinase, succinic acid with the release of coenzyme. Water is added and this step is not given, I think. Okay, succinyl CoA becomes succinic acid. So this step is not very important, but this succinic acid becomes fumaric acid, and succinic dehydrogenase is the enzyme that's given here, and this is oxidation 5. So this oxidation helps in the formation of what? Fumaric acid, and no, oxidation, removal of that hydrogen, succinic dehydrate. So this enzyme, please remember very carefully, this enzyme we will come across in the ETC, electron transport chain. Okay, where is it? It is one of the component of the electron transport chain. So where all these is happening? Whatever I'm saying, that biochemical pathway, where is it happening now?
Between the.
In the matrix of mitochondria. All of you please remember, where is this happening? So you know that mitochondria is like this. So this is a folded. So this is the cristae. Okay, and you find the matrix here. So where is the matrix? Matrix is present here. So whatever I'm doing yellow, that is the matrix. So here is the matrix. It's happening in the matrix. So what is this outside? Outside is called as outer membrane. So this is cristae, and the space between the outer membrane and perimitochondrial space. Okay. So now perimitochondrial space. So this succinate dehydrogenase is present towards the perimitochondrial space. So that's why this component is important to understand the electron transport. So where electron transport is happening? In the perimitochondrial cells. Why it happens? So that's what we will understand. So now here, till 4 oxidation. First in glycolysis, second in conversion of pyruvate to coenzyme A. Third is when?
Conversion of acetyl coenzyme.
When is the third? If you have not written, none of you wrote. Every time dehydrogenase is the enzyme there.
Citric acid to oxalo.
Nobody wrote in your book? Online students?
Then oxalose.
Succinyl
Acid.
Nobody has written. So this is an important concept here, important step. Why this is an important? Because here substrate level phosphorylation 3 takes place, where 2 had taken place.
in the glycolysis.
When in glycolysis, substrate level phosphorylation. This is how the questions will come. This is how the NEET questions will come.
Phosphoenol pyruvate to pyruvate.
Which of the following is not a substrate level phosphorylation? Conversion of phosphoenol pyruvate to pyruvic acid. That's the second one. Which is the first one?
First one is fructose 6. No, triose, but it's phosphate to triose.
My dear, substrate level phosphorylation means formation of ATP, not utilization of ATP.
So it is 1,3 bis phosphoglyceric acid to 3 phosphoglyceric acid.
Yes. 1,3 bis phosphoglyceric acid, 1,3. So whatever the phosphate group present in the first carbon is removed and ATP. It is given to ADP to form ATP. That's the first substrate level phosphorylation. Second substrate level phosphorylation is conversion to the ATP. Third substrate level phosphorylation is succinic coenzyme converted to succinic acid. So, and ATP there, inorganic phosphate, and coenzyme A, when it is removed, lot of energy is released, and this energy is utilized to convert ADP and IP to ATP. So this is the third level component, this one. So this succinic acid is converted to fumaric acid. This is an important step where the utilization of the enzyme, that is succinic dehydrogenase. This is not alone an enzyme. It is also helps in the transfer of electron. Okay, so this you keep it in mind. So this succinic dehydrogenase has an imitator which acts like a succinic dehydrogenase. So that's called as competitive inhibitor, which look like a succinic dehydrogenase only, which is that malonic acid. This is an exclusive NEET question.
Is it malic acid? No.
No, malonic.
Malic acid is different. Malic acid is an intermediate product that's formed in CAM plants.
Yes.
This is malonic acid. So malonic acid is the acid that imitates a succinic dehydrogenase enzyme.
Yes.
Okay, so this malonic acid, sorry, not the enzyme, substrate. So therefore, malonic acid acts like succinic acid, and therefore succinic dehydrogenate, if at all malonic acid is present in your body, you die. Why? Because the succinic acid cannot bind to succinic dehydrogenase. So succinic dehydrogenase will bind to malonic acid. So when it binds to malonic acid, what happens? The further formation of ATP is not happening. Oxidation would not happen. It causes suffocation, breathlessness, hypoxia. So all these would occur. Are you understanding? So succinic acid imitator is malonic and this is called as competitive inhibitor. In biomolecules, this is a exclusive point I have written here. So succinic acid is converted to fumaric acid. Fumaric acid is converted to malic acid. And this is an very easy one. So there it is hydration reaction. So this is hydration. Hydration is giving the water. So next is another oxidation. Total oxidation, keep counting. So here there is sixth oxidation. So this is malic acid is converted to oxaloacetic acid. That is the cycle, right? So this malic acid is converted to oxaloacetic acid, and here also NAD is converted to NADH. So here FAD is formed. This is also oxidation, but here instead of this important point, I told you, no, instead of NAD, FAD. What is FAD? What is the full form of FAD?
Fluorine
I told you all these are coenzymes derived from vitamins.
Yes.
Nicotinamide adenine dinucleotide is NAD. FAD? Flavin adenine dinucleotide, riboflavin, derived from vitamin A. Got it?
Yes.
So they are derived from vitamins. So that's why vitamins important for your body, which will enhance the cell division, respiration, all these.
Like how much is one FADH2 like?
That's what I'm coming to, that balance sheet now. So balance sheet of Krebs cycle. So, oh, I have written beta oxidation here. So here 6 NAD is formed in Krebs cycle, 2 FAD forms and 2 ATP forms. That is one substrate level phosphorylation. 6 NADP means 3 places into 6 into 2, 6. So one place FAD, that is 2. So total 6 NADH and 2 FAD and 2 ATP. So did I write the complete ATP formation? So I will write that ATP formation in the end. So what is the balance sheet of glycolysis? How many? What is the balance sheet of glycolysis, guys? Online students, let them tell.
Ma'am, 2 NADH and 4 minus 2, that is 2 ATP.
Okay.
2 NADH is right. How many ATP?
2 ATP.
Net ATP. 2 or 4? 2
4.
Wait. How many you consume? Investment is how much?
4.
How much is consumed?
2.
Investment phase. 2 or go to glycolysis. How much you consume? All of you go to glycolysis. How much you consume? Wait, like they will do it. Wait. Meda. Vihan, today you are not talking at all. Why? Meda, you were very active in the beginning. What happened to you, dear? Are you there in the seat or not? Meda. Wait, why are you giving commentary?
Cricket commentary.
Meda? Vihan?
Yes, ma'am.
Okay. So now come on, Vihan, tell me.
Ma'am, could you repeat the question?
Give the balance sheet, that's all, of glycolysis. Go to glycolysis.
Yes, ma'am.
Hmm.
Ma'am, 2 NADH plus H plus is
When do you get to NAD? When that first oxidation takes place?
ma'am.
When that first oxidation takes place?
Ma'am, and glyceraldehyde 3-phosphate breaks down to give
Two?
to give 1,3-biphosphoglyceric acid.
Acid. Aldehyde, remember when aldehyde becomes acid by the removal of hydrogen. So there the first oxidation takes place. Okay, that is NADH. So 2 NAD. Why 2 NAD? Why not 1?
There are two molecules.
After cleavage of which substance will get cleaved?
Fructose 1,6
Fructose 1,6 biphosphate is cleaved into
triose phosphate.
triose. See, hexose is cleaved into triose. Okay, so then every time we are getting 2 and hence we are writing 2. Okay, 2 NAD. So that's the first oxidation. So which are the 2 places you get ATP? 2 molecules of ATP each in 2 steps. SLP, that is substrate level phosphorylation 1 and substrate level phosphorylation 2. Which place?
Ma'am, when 1,3-biphosphoglyceric acid breaks down to give phosphoglyceric acid and
Yes.
what is it? Phosphoenol pyruvate breaks down to give pyruvic acid.
Pyruvic acid in the end ones, and 1,3 becomes 3. That's all. That is the one. So how many ATPs are formed each time?
Ma'am, at each step or in total?
4 in total. So how many is invested?
Ma'am, 2.
2. So what is the net?
2.
2.
2 ATP gain.
Got it?
Yeah.
So how many NADP?
2.
2. So 2 NADP, 2 ATP. And here
No, NAD.
NADH. Yeah. So here 6 NADH, 2 FAD and 2 ATP. So which are the 3 places oxidation has taken place here in Krebs cycle? Okay, let me give you the oxidation. So that is this is the best picture. So depiction, if you write everything in the cycle, you need not describe in the form of steps. So let me show you that particular cycle here. So becomes easy for you. Hopefully you can see everything in this cycle. Yes, this cycle has everything. So here the first oxidation. So I have written here oxidation 3. Can you see here oxidation 3?
Yes.
I have written here oxidation 4 and oxidation 5. Where is oxidation 5? This one.
FADH.
Succeeded to fumarate.
Correct. So oxidation 3. Where is pencil? Let me write it with that pencil kind of thing. Here. So this is 3. This is oxidation 4. I can type also. But writing becomes easy. So 4 and this is oxidation 5 and this is oxidation 6. Okay. So now total oxidation happens. So 1 has happened in glycolysis. 2 has happened from pyruvate to acetyl-CoA. 3 here. 4, 3 when? Isocitrate to oxalosuccinate. So now glutarate to succinyl-CoA. So now succinate to fumarate 1 and malate to oxaloacetic acid in the end 2. So every time, so there are 6. And how many ATPs are generated here?
2.
So there is 2 ATPs, that's 1 SLP. So when succinyl-CoA to succinate. So which is the enzyme there? Thiokinase. Kinase is the enzyme, has to be there. If you do not know anything, do not get into your mind anything. If you write kinase, 100% marks has to be given. So remember that point, kinase. Thiokinase is the enzyme. So what is the balance sheet with respect to Krebs cycle? So 2 ATP, 6 NADP and 1 FAD. Got it? So this is the balance sheet. Now, what is the significance of this Krebs cycle? So what process? So now when we talk about photosynthesis and respiration, we say that respiration is a catabolic process and photosynthesis is an anabolic process. Why do we call it a catabolic process? Because the organic compounds break down here, right? So this is what is the other significance? So it's a common pathway of oxidative breakdown of carbohydrates, fatty acids, amino acids, everything break down and gets into the Krebs cycle. So here, alpha ketogluteric acid produces an important amino acid glutamate.
Yes.
The intermediate products, I'm talking about the usage of every intermediate product that's formed during Krebs cycle. Now, breakdown of the pyruvic acid is happening step by step to release carbon dioxide. So now in this release of carbon dioxide, what is formed? From alpha ketogluteric acid, what is formed? Glutamate. Glutamate is a glutaric acid. Glutamate or glutaric acid is a amino acid. Aspartic acid is a formed from oxaloacetic acid. Fat and lipids form fatty acid and glycerol during beta oxidation. During beta oxidation. They form fatty acid and glycerol, which is further used for building up process of many things in our body. Acetyl coenzyme produce 2 carbon compounds for the synthesis of fatty acids, cutin in plants, aromatic compounds, isoprenoids, all about in plants. Since we are talking with respect to respiration in plants. So these are all required for the formation of chlorophyll, carotenoids, terpenes, gibberellins,
Yes.
all these in the plants. Succinyl-CoA, okay, forms cytochromes and phytochromes. Cytochromes in other organisms, phytochromes in plants. So through Krebs cycle, though Krebs cycle is involved in catabolic process, breakdown process, it's involved, their intermediate products are involved in the building up of many biomolecules. Are you understanding? So therefore, it is not completely catabolic process. It's called as amphibolic process. So though there is a breakdown of the substances, those broken down substances are used to build chota some other substances.
Yes.
Are you understanding? So hence, Krebs cycle is called as amphibolic pathway. So this is a proper subjective 3-mark question. Krebs cycle is an amphibolic pathway. Discuss or elaborate, enumerate the points, anything they could give. Now, how many places did you see here the release of carbon dioxide? Can you tell me where all the carbon dioxide decarboxylation has taken place? So how many molecules of carbon dioxide has been given out so far?
2. No, 3.
Where all? Which are the places? Yes, online students. I want answer from online students. Medha is trying to unmute, but it's not happening. I don't know what is I didn't get the picture correctly. What has happened? So where is the first carbon dioxide is given out? Is it? You people forget that linking. Okay, glycolysis and Krebs cycle, which is that linking? Pyruvate to coenzyme. That's a linking reaction.
Yes.
So in this linking reaction, carbon dioxide is given out. Can you see there? So the first carbon dioxide is given out there. So total, how many molecules of carbon dioxide so far is given out? You have to count now. Okay, one only. Where to? Where to?
Oxalosuccinate to alpha ketoglutarate.
Where is to? This picture I have taken, I have exclusively taken and altered also by typing and all these to give you the complete picture. Where is the second carboxylation, decarboxylation?
Oxalosuccinate.
Online students. Oxalosuccinic acid to alpha glutaric acid. Second, and again alpha glutaric acid to succinyl CoA. Third, and then again where?
So 3 is produced.
So how many carbon dioxide decarboxylation taken place? So once during pyruvate, and here decarboxylase is used. So that is decarboxylation taken happens there. And second decarboxylation happens, I have written here every point. Second decarboxylation here, see. Yeah, decarboxylation 2, I have written here 2. Okay, so that is oxalosuccinic acid to alpha glutaric acid. Where is the third decarboxylation?
Alpha glutaric acid to
Succinic acid. So here decarboxylation 3. Can you see here?
Yes.
Decarboxylation 3. So total there are 3 decarboxylation, 3 carbon dioxide is released in this complete processes, 2 processes. Okay, so this is the balance sheet. Hopefully you have understood so far this much clearly. So in the next class, let us take up the electron transport and balance sheet correctly. And what else is there after electron transport and the balance sheet?
Respiratory balance.
That's all, right?
Amphibiolic.
That we discussed just now. So-
Respiratory quotient.
Quotient, that is respiratory quotient is a one simple concept, and balance sheet, and so hardly I require 1 hour. So another 1 hour, let's take up complete question answers from this, because this is involving a lot of reactions. So if you do more and more questions, the gap will be filled.
Yes.
Like how we did for photosynthesis to a greater extent. So let me see how you people do. I want everyone take up this Cayman test. So photosynthesis and breathing. So your knowledge becomes better. So even though you do wrong, that's absolutely fine. You will get to see the solution.
Yes.
And when you do, believe me, when you do some mistakes and then you learn the correct answer, your memory retention will be better. Right?
Yes.
So when we do, suppose when we are pronouncing something, if we do mistake, or when we are writing something, we do mistake, if somebody corrects, or if we get to see the solution, we tend to remember that better way. So that's why I suggest you people to take up this Cayman without fail. So I want everyone today to it. So it will be open for a week. So kindly take it up. It's for your own good. Especially photosynthesis and respiration requires a lot of biochemical reactions, and it's better if you take it up. So once it is done in the next class for 2 hours, let's do it. And last hour I'll do for finish plant growth and development. So we are done with that complete portions of plant physiology. Okay, so I hardly require 1 hour, 20 minutes or that like that. So it has nothing. Very, very easy. You can connect it to that very easily. So only thing is it's 100% memory. Why? Because all about phytohormones. So I'll give you a table. So you people write the table in the chart paper, hang it up. So unit 1, 100% memory. Unit 1, biological classifications, names of plants and names of animals. Right? So that's 100% memory. And mineral nutrition, we are done with that, right? Have you done with that?
Yes.
Yes.
We did it?
Yes.
Okay, so in that it's more of memory because you learn all about all the minerals and their deficiency diseases. So this is 100% memory, and last chapter, chemical coordination, all about the hormones and their diseases, functions. That's also almost 98% memory. So these memory chapters, no teacher is required, honestly speaking. So teacher is required only to memorize, bring it into your mind, that's all.
Yes.
So these chapters, yes, required. So those chapters, you have to make a chart paper. So on the chart paper, make the tabulation and keep it up, hang it up somewhere wherever you are moving around. So that keep seeing that every day. So which will help in the better memory retention. So that's all I can say.
5 are there, right?
5 different chapters.
No,
Then
in the plant growth and development.
plant growth and development, the phytohormones. So these are growth regulators. They are called as growth promoters and growth inhibitors. Growth promoters are auxins, gibberellins, and cytokinins. Growth inhibitors are ethylene and abscisic acid. So totally there are 5 different hormones which we will be learning. So hardly I require 1 hour 20 minutes to do that. So in the next class, let me complete this also and cover that, so that we will be done with the plant physiology. So then we can take up another very, very lengthy and very interesting for students. I know that is all about your nervous system, and we'll be learning about your big boss in detail. Okay. So thank you all of you. Stay safe.
So how many chapters left?
So next class onwards, then we will be left with 2 only after the next class. Okay. Thank you.
After that, like full test.
Ma'am.
Yes, after that, we'll break it up and give. There's no point. You people do not study if I give full book.